you know, skip through what's probably not so important. So, methods of gene therapy, there are three methods. One is retrovirus, which, or there are three viral approaches, let's put it this way. One is a retrovirus that infects dividing cells. The second is the adenovirus that can infect both dividing and resting cells, but the adenovirus itself invokes a pretty hefty immune response. And the herpes virus is much bigger and very pathogenic, and that's why people are somehow kind of a little afraid of it. And you'll notice when we're talking about COVID, we're actually delivering genes to cells for our immune response, hoping to generate an immune response. So it's not so much different than what we would do for gene therapy for radiation oncology. The virus that is being used not for the Moderna or the Pfizer, but the AstraZeneca vaccine actually is adenovirus, and they're putting the spike sequence in the adenovirus to generate immune responses. Um, the things that we've tried to do from a um, radiation oncology perspective are to add in suicide genes, to maybe add in a virus that would be cytotoxic for cells that are deficient in P53 or RB. There are some examples that have been done. There are cancer vaccines where we add an antibody to RAS. And again, I would say COVID fits into this kind of category. It's not cancer, but they're making antibodies against particular proteins, tumor suppressor gene therapy, and then the radiation inducible cytotoxic agent, which is one of the ones that's been on the boards. In all of these mechanisms, they usually involve some sort of um, enhancing immune responses. This has become very important to us clinically, but right now we're doing mostly immune enhancement directly with radiotherapy. There are approaches to be able to generate several different immune responses against the tumor itself. One of the ones that has been is very complicated, but people have done is where you generate uh, anti-tumor immunity in culture in with using the patient's own cells and put them back into the patient. Um, this is one model that's been on the boards in the past. It is a kind of suicide gene therapy approach. It was used for glioblastomas, not so successfully. It has been touted as being useful in a couple of other systems. The idea is that you give systemically, this should say systemically, ganciclovir. This is a non-cytotoxic drug, so it has very little side effects for the patient. You then add to the patient the herpes simplex. You infect with herpes simplex virus, the herpes simplex virus genome, which contains the thymidine kinase gene. This now goes in. The thymidine kinase takes the inactive ganciclovir drug, makes it be phosphorylated, and makes it be toxic for the cells. Um, there are a couple of things to note. In wait, let me show you this image. It might be a little bit better, and then we can go back to that other slide. So here's kind of the image. They don't really talk about the drug ganciclovir, but that's what the drug is they're using as their example here. So you take the herpes simplex virus. It has its own thymidine kinase in it. We also make our own thymidine kinase, but it will not uh, change this prodrug into an active drug. So the virus goes in, it makes the thymidine kinase, RNA makes the enzyme. When the person has received the prodrug, the enzyme goes in, phosphorylates the prodrug, makes it be toxic, and it kills the cancer cell. The interesting thing about this particular drug, the ganciclovir version, is that it is also, it's not just toxic for itself, but it crosses the gap junctions and can be toxic for neighboring cells. This is called a bystander effect. We will hear later in the low dose literature that a bystander effect is also very different in the radiation field, but the concept is you have a cell that is affected, and that cell, or that is in this example, has the drug, but the neighboring cell, which never received the drug, dies. And for radiation, it's going to be the cell next to the one that was irradiated has a response. Okay. Uh, the way in which we get specificity for tumor here is the ganciclovir goes throughout the entire body. It's given systemically. There's no question about where it goes. But the herpes virus infects the tumor. And in fact, in the case of GBM, the virus was actually given during surgery to be able to carry out this function. So the cells that have both ganciclovir and thymidine kinase die, but either one alone is not toxic. And there's this bystander effect where material passes through the gap junctions to neighboring cells, and there is an immune response that is induced. So this is one, this has been mentioned on the boards before, and it is kind of the mechanism of action of most ganciclovir therapies. Okay. The second one that has been, it was pioneered by Ralph Voxelbaum at University of Chicago, is this mechanism where he put a toxic agent, in this case he used a natural toxin, TNF alpha. TNF got its name because it causes necrosis of tumor cells. So the idea was you have this driven by a radiation inducible promoter. He used the EGR1 promoter. You package this into an adenovirus, you put it into the patient, and wherever the cells are irradiated, you get turn on of additional TNF alpha more than what there is produced in the normal host cells anyway. So, so the advantages to this are that you're using a kind of natural agent. You're not giving the cell any kind of toxin that it hasn't seen already. The second advantage is that while this virus goes all over the body, the radiation only goes to certain areas. And so therefore where you irradiate, that's where you get the specific expression, and that gives it some specificity. The concept was you give these to patients, you administer it, you treat with this agent, you now expect to get therapy. We have improved patient outcome in the treated. The control has poor patient outcome, and the net result is expressed. And this is just another diagram showing the adenovirus, the radiation treatment, and the killing. Or as Ralph argued, TNF can also be a pretty powerful radiosensitizer. So that was another mechanism that was proposed. And indeed, in vitro studies, here's our old friend, the tumor growth delay. The tumor was treated. Here's the control where the tumor's growing. Give 50 gray of radiation, and it does decrease some. But give it the radiation plus, and give it the TNF alone, and it goes down a little bit, but not much. But give it the TNF driven, and give it the TNF with the adenovirus construct, it goes down not much. Give it the TNF adenovirus construct with 50 gray, and you can see you get really a pretty significant growth delay in the tumors. So this was quite exciting. There was a lot of discussion of other approaches for gene therapy. One of them was called radiation-enhanced viral oncolytic therapy, or REVOLT. This is where a mutant virus came in and would alter certain functions within the cell. That has not panned out to be very successful. There was tumor suppressor gene therapy that was given, and the idea was that you give wild type P53 to cells that are already deficient in P53, and you get radiosensitizer function. The problem was this was only going to work for those that have a mutant P53, not those that have, say, a virus that suppresses P53. In the end, it did work in a phase 1 clinical trial where it inhibited tumor growth, but not enough to have it be change clinical outcome. And so therefore that was sort of put down the tubes as well. There was effort using cytolytic viruses for P53 specifically. The virus would grow only in P53 deficient cells and kill them. This was not very effective either. Here are some constructs that have been attempted showing that concept of radiation induction. There are some that where an HSV mutant was used, adenovirus mutants were used. So many were tried in many different tumor types. There was always some sort of impact, but it was never enough to make it be useful clinically. And so people, at the end of the day, I think most of this was just we sort of gave up. I will mention cancer vaccines. One idea was to create some sort of virus that would stimulate immune responses, not just enhance TNF expression, which is what Ralph had tried to do, but to stimulate IL-2, GM-CSF, or gamma interferon expression. The problem was they weren't sure what would constitute a good immune response there. It wasn't that good to induce all of these. Finally, people gave up and they went to a regular immunization using human papillomavirus to protect against cervical carcinoma. That's had a lot of controversy associated with it. It only works against certain subtypes, but it works against the main subtypes that are most important for human cancers, and therefore it has been somewhat successful. The idea of this kind of immune therapy with cytokine delivery is shown here, where what you do is you have this virus particle, it stimulates immune responses, it contains within it, say for instance, GM-CSF, which we've learned is a radioprotector actually, right? Or radio restorative agent for bone marrow. But in this example, they're using it to help enhance the ability of antigen presenting cells to present antigen to cytotoxic lymphocytes, which it also does.
I thought the winner does.
I'm sorry. Okay, I'm going to just keep going. So here's the antigen presenting cell getting stimulated by the GM-CSF that's produced by the virus particle. And this has been a form of gene therapy that has been quite successful. There are other types of gene therapy where people have tried to use radioisotopes to target particular tumors. And we talked about that a little bit when we talked about brachytherapy. So I think that's the more relevant thing. And so let me just end this. There are these molecular approaches that are being used to bring gene therapy into the treatment arena. There are these many approaches that tend to use different kinds of viral vectors and different kinds of suicide genes, and cancer vaccines are also being attempted.